So now that we know a bit about the divisions of the nervous system, it's time to actually delve deeper and have a look at the microscopic structures of the nervous system. And the functional and structural unit of the nervous system, the thing that makes it all work, the smallest component that makes it work, is called the neurons. So we're going to be looking at that in this video here. So let's have a look at the basic structure of a myelinated neuron. Now there's three parts to a neuron. So one is the dendrite, and the dendrite are like little antennae that are coming directly off the cell body, and these are going to be little receivers of information. So it may be information about the external or the internal environment. It may be receiving information from another neuron, for example. Now, if we look at what the dendrites are actually doing, they're coming directly off the cell body and they're actually splitting, and this is a structure, an arrangement rather, called arborization, and arbor means tree. So they're actually splitting into smaller and smaller branches, just like a tree would be. So they demonstrate arborization. Now, if we look at the cell body here, it's also known as the perikaryon, and that's going to be containing a lot of different things. It's going to be containing the nucleus, and the nucleus is important for housing the genetic information, so the information that the neuron needs to actually run all the processes of the neuron. And of course, this is where the DNA is housed. And it also houses organelles, and there's one specific organelle that's referred to as Nissl bodies. Nissl was Dr. Nissl, named after Dr. Nissl, and he was a German psychiatrist, and he saw these things under the microscopes. And what Nissl bodies actually are is an organelle, and the organelle is called rough endoplasmic reticulum. And rough endoplasmic reticulum is studded with little protein-producing machines. And there's a lot of different proteins that need to be manufactured for the neurons. But arguably the most important of those is the neurotransmitters. So the chemicals that actually get released by the neuron to communicate with other structures. So that gets manufactured in the rough endoplasmic reticulum, which is seen under the microscope as a Nissl body. Can be a bit confusing. Try and get your head around that. Now, the next thing we're going to look at is this, and this is called the axon, and the axon is the conducting part of the cell. And the information that travels through the axon is encoded as an electrical signal. And the electrical signal is referred to as an action potential. And we won't be looking much more about the physiology of the action potentials in this unit, but you will, however, in human structure and function. So this whole area is known as an axon. Now you can see that the axon is covered by this grey substance here, and that's called the myelin sheath. Now myelin is a material that's made up mostly of lipid, mostly of fat-derived products, and a little bit of protein as well. And it's an insulating material. And the whole point of having this myelin sheath is that it's going to insulate the axon, so that's almost like a wire from the neuron, and it's going to speed up the transmission of these action potentials, of the electrical signals that actually flow through the axons. And what you can see is that occasionally this myelin sheath comes to a bare area, and they're little nodes, and they're going to be known as the nodes of Ranvier. And Ranvier, I'm not pronouncing that to be pretentious. Ranvier was actually a French physician who identified these little nodes of unmyelinated axon. And so that's why we term it the nodes of Ranvier. And because they're not covered with myelin sheath, this is areas of the axon that can actually conduct electricity. And so what happens is that the action potentials actually jump from one node of Ranvier to another node of Ranvier. So it speeds up conduction. So that's two things that we've spoken about that speed up conduction. One is the fact that it's myelinated, and the fact that we have these little gaps in the myelin sheath allows electrical signals to jump between them. Now on the end, what you can see here is the axon terminals, and sometimes referred to as the synaptic bouton. And again, that's a French term, and it means buttons. Why? Because when the first people looked down at these little axon terminals, they thought these looked like little buttons. So the French named them the synaptic bouton. So, bit of French for you. Right, moving on. So hopefully you've got a reasonable idea of what a neuron actually looks like. And this is a myelinated neuron. Why? Because it's got a myelin sheath. If it didn't have a myelin sheath, we'd refer to it as an unmyelinated neuron. And there's lots of different types of neuron. The first that we're going to look at is a bipolar neuron. And this is bipolar because it's got two poles. So it's got one dendrite and one axon. And so what happens is that the action potential is going to start in the dendrite portion, the dendritic portion of this bipolar neuron, and it's going to travel through the cell body and down the axon to the axon terminals. And where you'll find these bipolar neurons, it's actually quite a rare type of neuron, is in the neurons for special senses. So what might happen, let's say this is these dendrites are in the retina, for example, then light will hit the special receptor that detects light. It will propagate an action potential from the dendrite through the cell body, down the axon, and towards the brain. So, not a common type of neuron. The next one we're going to look at is a pseudo-unipolar neuron. And what you can see here is that from the cell body, it looks like it only has one axon. And we've got two parts of these axons. The peripheral axon, which as the name suggests, is further towards the periphery, and the central axon, which is closer to the central part of the nervous system. Now, pseudo-unipolar neurons don't technically have dendrites, although they're labelled here. These are not true dendrites, because true dendrites will actually come off these cell bodies, so not true dendrites there. But what will happen with the pseudo-unipolar neurons is that the action potential will start to be propagated up here in these peripheral terminals of the peripheral axon, and it will start to move through this axon, through the central axon, out towards the axon terminals. And where we'll find this type of neuron is in our sensory neurons, our general sensory neurons. And for example, let's say that these peripheral terminals that you can see here were exposed to a painful stimulus. Well, what happens is that they start an action potential that gets propagated through the peripheral axon, down the central axon, towards the axon terminals, and down here, these axon terminals will communicate with the central nervous system, with the spinal cord. So that's how that works, but it doesn't necessarily need to be pain. At the end of these little terminals, peripheral terminals, there's little sensory receptors. So instead of pain, we could be talking about light touch, crude touch, pressure, vibration, all these sorts of things. Heat, cold, etc. Now, a third type of neuron that we can look at is a multipolar neuron. And it's called multipolar because you can see it has multiple dendrites coming directly from its cell body. But it still only has one axon. And so what happens is that an action potential from here, if the dendrites are stimulated enough, will be generated at an area called the axon hillock. And the action potential will be propagated right down to the axon terminal. And you'll find this type of neuron in two places. you'll find them being our motor neurons and some being our interneurons. So our motor neurons are neurons that are going to stimulate muscle, especially skeletal muscle. And our interneurons are going to be the neurons that are going to talk to other neurons. So with a motor neuron, this cell body is going to be within the spinal cord itself. And so it's going to receive a signal through these dendrites saying, you must fire, you need to send a message to the muscle to fire. So that action potential goes down through the axon and will tell a muscle down here to start firing. With an interneuron, that's a bit different because the interneuron is sort of the neuron sandwiched between two other neurons. So a neuron will talk to these dendrites, an action potential will be generated, it will send an action potential through the axon, and it will talk to another neuron on the other end of these axon terminals. So that's termed an interneuron. So now that we know the different types of neurons and the structure of neurons, we need to look at how neurons actually talk to each other. And they talk to each other through what's known as a synapse. And so what happens, we can look at this. This is just the axon terminal. So that's just a big version of what we've been seeing here, these synaptic boutons. And what will happen is an action potential will arrive at the axon terminal, and we have these calcium channels here, and these calcium channels are voltage gated. That means when this voltage arrives down here, calcium will take an influx into this area here, which is known as the presynaptic terminal. What happens is that calcium flowing into the presynaptic terminal will cause these little synaptic vesicles to bind to this area, which is called the presynaptic membrane. What happens when the synaptic vesicles bind to the presynaptic membrane is that neurotransmitters will be released, and these neurotransmitters have previously been housed within these vesicles. So when the neurotransmitters get released into this area, called the synaptic cleft, they diffuse down towards this membrane, and this membrane is referred to as the postsynaptic membrane. And it's important to note here that this membrane could be on lots of different places. The postsynaptic membrane could be on a skeletal muscle, for example. It could be on another neuron, or it could be on a gland, for example. So this postsynaptic membrane could be on lots of different places. So what happens when the neurotransmitter starts to bind to these receptors that you can see here? The receptor actually opens. And so what happens when the receptor opens is that these ions will actually flow through it. And what happens is that these ions could be stimulatory or inhibitory. In this case, it's stimulatory because it's sodium. And so that will flow through down into, once again, it could be another neuron, it could be a muscle, it could be a gland, and it will stimulate that neuron, muscle, or gland. So that's how neurons talk to their target structures. And so let's have a look at the different types of synapses we could have. We said we could have a synapse of one neuron with another neuron, and we simply just call this a synapse. Now, if this synapse happens to be on skeletal muscle, then this synapse is known as a neuromuscular junction. If this synapse is on a gland, for example, we know this as a neuroglandular synapse. So they're the three different types of synapses that you need to know. So now we've talked about the structure of a synapse. We need to go back a little bit because I talked briefly about the myelin sheath. And there's two different cells that myelinate axons. One is called the oligodendrocyte that you can see here. And the oligodendrocytes are going to myelinate axons in the central nervous system. And this is what an oligodendrocyte looks like. The unique feature of an oligodendrocyte is that one cell can actually myelinate several axons. And this is implicit in the name because we've got few, that's oligo, so that's few, dendros. So that's a process. So what we can see is it's got several processes. So it can myelinate several different axons. But this only occurs in the central nervous system. And you can see here in the picture in the bottom left, here's an oligodendrocyte, and here's its little processes coming down to several different axons to myelinate them. Now the myelination in the peripheral nervous system gets done by the Schwann cells. The difference between the Schwann cells and the oligodendrocytes is that the Schwann cell can only myelinate one axon. And so you can see here's a Schwann cell, here's another Schwann cell. The nodes between them, the unmyelinated nodes, is referred to as the node of Ranvier. You can see the nucleus of the Schwann cell here, and you can see the myelin sheath that the Schwann cell is actually producing. And so you can see that this exists in the myelinated axon, and you can see that Schwann cells exist in the unmyelinated axon. However, the difference is that those Schwann cells aren't actively secreting the myelin. And so, although the Schwann cells, these axons are bare, they're not actually surrounded by myelin sheath. Instead, they're surrounded by a thin layer of cytoplasm from the Schwann cell. So, please remember that. Schwann cells do the myelinating in the peripheral nervous system. And so you can see here on this electron micrograph, you can see this axon here, you can see this large structure, this is the Schwann cell, and you can see this thick layer here, and this is the myelin sheath. Now this myelination actually has a lot of relevance to when we look at the neural structures grossly, because we can see two different types of matter. We can see white matter and we can see grey matter. And this is just a cross-section, a transverse section through the brain. We call it white and grey matter because that's how it looks on a fresh neural specimen. And so here you can see the white matter, here you can see the grey matter. The question is, what makes white matter white and what makes grey matter grey? Well, the white matter is white because it contains lots and lots of myelinated axons. And if you remember back to the first slide, I said that myelin is made up mostly of lipid, so mostly of fatty tissue, and it's actually a cholesterol-derived tissue, and we often talk about the negative effects of cholesterol, but in fact it's an essential precursor to myelin. And if you open up a body and look at fat, it looks white. And so this is why myelinated axons make up our white matter. Contrast that to our grey matter, which consists mostly of cell bodies, dendrites, and synapses. Now, cell bodies, dendrites, and synapses are not myelinated, therefore it starts to look grey. And we'll look a bit more carefully into how this looks, how the white matter and grey matter look on different imaging modalities as well, because it's not necessarily clear-cut on different types of imaging. But we'll do that at a later stage. So the last thing we're going to look at is the histology of an axon, and the way we're looking at it here is in longitudinal sections. So we've cut down the length of the axon rather than across it. If we cut across it, it would be a transverse section. If we cut down the length of it, as we have here, it's a longitudinal section. So I'm just going to talk you through what we can see here on this histology slide. So the first thing is this long cylindrical structure, and this is of course the axon. We can see several nuclei surrounding the axon, and they're going to be Schwann cells. So we're in the peripheral nervous system here. What you can see here looks like a space, but it's not actually a space. The area that you can see here is actually pointing to the myelin sheath. The reason it looks like nothing is because myelin is mostly lipid. It's mostly, as we said, cholesterol derived. And so what happens during the histological processing, lipid actually gets lost. And it's similar to when we looked at the yellow bone marrow and looked at those adipose cells, and they looked empty. Well, they're not actually empty, they're just filled with lipid, and that's the same as the myelin sheath. And it's rather interesting actually that myelin sheath has the abbreviation MS, because multiple sclerosis actually attacks the myelin sheath. But multiple sclerosis attacks the myelin sheath in the central nervous system. And so obviously this axon that we can see here would be spared, because we can clearly see it's in the peripheral nervous system because we can see Schwann cells. Right, moving on. You can see these little unmyelinated portions of the axon here, and these are going to be our nodes of Ranvier. So if we have a look at the histology of an axon, but this time in a transverse section, what you can see is an axon, and so as we said, we've cut across. We're in cross section now instead of in a longitudinal section. We can see our Schwann cell nuclei. So again, it must mean that these axons are in the peripheral nervous system. And what we can see is this gap between the axon, which is here, and the Schwann cell there. So this would be the area that the myelin sheath would have occupied. And a final note on nomenclature. And nomenclature is just a fancy science term for the way we name things. And so a collection of cell bodies in the central nervous system is called a nucleus. And don't get confused, because we also have, of course, cell nuclei, which is an organelle, okay? This is not the same thing. A cell nucleus and a neural system nucleus is not the same thing. So a collection of cell bodies in the central nervous system is referred to as a nucleus, whereas a collection of cell bodies in the peripheral nervous system is known as a ganglion. A collection of axons in the central nervous system is known as a tract. However, a collection of axons in the peripheral nervous system is known as a nerve. So hopefully this clears a few things up for you. And in the next couple of videos, we're going to look at the difference between the central nervous system and its components and the peripheral nervous system. Thank you.